Hello, I'm Sam Yeaman. I'm a professor at the University of Calgary, and I'm here to talk to you today about how is information coded in our DNA. Now, you may have heard that the fundamental code of DNA is written using A, C, T, and G for the nucleotides adenine, cytosine, thymine, and guanine. Now, this is of course the fundamental code, but there's actually a lot more complicated stuff going on that also represents information within DNA. First, we'll just consider this fundamental code though. Basically, your genome consists of these very long strings of A, C, T, and G, which make up our chromosomes. And on our chromosomes, we have genes interspersed by other non-coding regions. So each gene is a coding region. There's a start and a stop part of the gene, and in between, the sequence of A, C, T, and G gives the cell the information it needs to figure out how to make a protein. Basically, the combinations of A, C, T, and G are read in blocks of 3 called codons, and each codon codes for a particular amino acid. And so by reading through these genes, the cell figures out what sequence of amino acids to build to make the protein that's coded for by that gene. So this is the fundamental dogma of molecular biology, and this is the real primary way that information is coded within your DNA as actual genes. But there are other more kind of subtle and interesting ways that information is also represented that we're still learning about. One primary way is that promoters are used along near genes. So there's flanking regions, nearby genes that are used to help turn genes on and off and tell the cell, this is a gene that we need to be running right now. And so basically, the sequence of A, C, T, and G in the promoter region, either upstream or downstream from a gene, can be used to recruit other proteins that recognize, oh, that's the specific sequence, and they stick to it. And those proteins then recruit the machinery that turns on the gene. And so having a promoter sequence nearby a gene is essential for actually getting that gene to work properly. And the letters A, C, T, and G in that promoter region are therefore primarily important for actually making that work. So you can say there's also information coded in the sequence of promoter regions because we need to have that sequence working for proper binding of the promoter proteins. We've also been very interested lately in science in something called methylation and epigenetics. Basically, we can have methylation marks added to DNA, which are these marks that are basically added on to different parts of DNA, and they are used to turn genes on and off in a different way. Epigenetic marks may occur based on responses to the environment. So if we experience very stressful conditions, that may tell our body to turn on or turn off genes in particular ways, and that may change our methylation status. Interestingly though, most methylation marks aren't actually passed on to the next generation, although some of them are. There's still a lot of work being done on epigenetics and learning about how methylation status represents a kind of parallel way of encoding information in DNA, but not kind of as primary as the actual sequence of letters in your genes. There's other really exciting developments that are being found now in DNA science, looking at how chromosomes are packed in the nucleus. Basically, the way that they're packed can affect the expression and how genes are turned on and off, and we're still learning lots about how this may potentially represent another source of information coding in the genome. Basically, though, it comes down to there's the first way of representing information is through the A, C, T, and G within your coding sequence, and then the secondary and tertiary ways of representing information are in various ways that those genes are turned on and off, that they're expressed and they're allowed to turn their information into proteins. So those are the two kind of broad categories, primary and secondary. And there's still lots that we have to learn. Very exciting time to be a scientist. I hope this has been helpful. I don't think it's a stretch to say that DNA is perhaps the most fascinating molecule we've ever found, and it's a really neat thing to have a chance to study these days.